Okay, photosynthesis. So in this section, we're just going to try to get a general idea or an overview of what's happening in photosynthesis. So photosynthesis is the conversion of light energy into chemical energy. And we can actually get the definition from the name itself. Photo actually means light, and synthesis means to put something together or to make something. And what are we making? We're making chemical energy, in this case a carbohydrate, from light. So we illustrate this in the form of this equation where we have carbon dioxide and water combined with light to give us a carbohydrate. In this case, we use glucose as the example, and oxygen. So we typically do use glucose as the example. So this is the balanced equation. If you kind of have to remember some chemistry there, know that this is a balanced equation, and memorize this, absolutely, because that question, put photosynthesis in the form of an equation is almost always asked on a test. So absolutely memorize that, okay? That's definitely step one. Now, I always think it's important to understand, big picture, real life stuff, what's going on. Now, we see here that one of the things that's produced in photosynthesis is actually oxygen. So this is why people make such a big deal when trees are cut down, or why people want to plant trees. Because by doing that, we're actually making, by planting trees, we're actually making the air cleaner because they're producing oxygen that we will breathe in, right? So that's just sort of a way to sort of fit that in. I think, kind of that's an important concept to understand, because up to now you're like, why, it's a big deal, like cutting the trees, I don't get it. Well, because of photosynthesis. So in this process of photosynthesis, there are two stages. We call them the light reactions and the dark reactions. The dark reactions, also known as the Calvin cycle, but we can also refer to it as well as the light-independent reactions, because they don't need light to take place. Now they don't necessarily happen at night. It's not like one happens during the day and one happens at night. It's just that these reactions don't need light to take place. And we're going to have a separate section on each of these, but I wanted you to get a general idea of photosynthesis before we sort of delve in. And this diagram here on the bottom, I really want you to keep this in mind as we go through. I like to do this, I like to show this to students, because when we're really sort of getting into the mix of things, going into the chloroplast and looking at what's going on down there, we lose the big picture of where the chloroplast is. Like, wait, where is this happening, and where does it go? So just sort of keep this little sequence in mind. We have our leaf. Inside our leaf is actually a plant cell. Sort of, looks like this, kind of square shape. Remember, plant cell has a cell wall, and we have some organelles in there in our nucleus. And we have some chloroplast floating around. Here's a chloroplast. And within our chloroplast, we have these things called thylakoids. Okay, and each thylakoid is like a little pancake here, and they're usually stacked on top of each other and interconnected, and we call actually a stack of these a grana, okay? Now, the thylakoid, and more specifically the thylakoid membrane, is where our light reactions occur. The stroma is this sort of fluid outside of the thylakoids, still in the chloroplast, and that's where our dark reactions, or our Calvin cycle, is going to take place. Okay, so here's sort of the overview, big picture. So next we're going to talk about the light reactions. Okay, so now we're going to talk about those light reactions. So the light reactions, like I was saying earlier, occur in the thylakoid, more specifically the thylakoid membrane. And I just want you to remember that a thylakoid is a disc that's usually, the thylakoids are usually in stacks, which remember we called grana, that are inside the chloroplast. And the chloroplast, as you recall, are actually inside a plant cell. Plant cells are inside of leaves. Just always go back to that big picture, because we are going to be getting into some pretty specific stuff. Okay, and I just want you to know that this space surrounding the thylakoids is called the stroma. It's this fluid substance that surrounds the thylakoid. And when we look at this diagram right here, which, by the way, this is the thylakoid membrane. This is a diagram of the thylakoid membrane. And when we're looking at it this way, you can consider this side of it to be the stroma, okay? So we are actually looking at outside of the thylakoid. And there actually is also a name for the inside, the interior space of the thylakoid membrane, and that is the lumen. So the lumen is the interior of the thylakoid membrane. Of the thylakoid membrane. Okay, so first thing I'm going to draw your attention to are these two guys right here, these two units right here. This one is called Photosystem 2, and this one is called Photosystem 1. Okay. And inside of these are various molecules, which include chlorophyll A and B, plus carotenoids. Okay, so I'm just going to put chlor, A and B, and carotenoids. Okay, the chlorophylls and the carotenoids are pigments. Pigments are molecules that absorb light. Okay, now chlorophyll B, as well as the carotenoids, we call accessory pigments. And it's chlorophyll A that's really the major player in the photosystem. So let me just show you how that works. So if we have light coming in, we have light hitting our photosystem here, what's going to happen is these accessory pigments, chlorophyll B and the carotenoids, they're going to absorb that energy, and they're going to pass that energy along to the chlorophyll A molecules. And when that happens, the electrons in those chlorophyll A molecules get excited and actually shoot out of here, shoot out of photosystem 2, okay? And they actually go into what we call an electron transport chain. So those electrons are passed from molecule to molecule within this thylakoid membrane. And I'm just going to label that right here, electron transport chain. All right. So as you can see, the chlorophyll B and the carotenoids, they were just more of the accessory. They absorbed that energy and they passed it along to the chlorophyll A. It was that chlorophyll A, though, that the electrons really got excited, and that's what shot out of the photosystem 2 and got things going along the electron transport chain. Now, what has happened here, though, is photosystem 2 is now missing electrons, and of course it wants to replace those. So this is where our water comes in the photosynthesis process. So let's say we have two molecules of water. What's going to happen is that's going to bind with an enzyme that's going to break this down to where we have 4 protons. H plus is how we signify protons, just in case you haven't come across that concept yet, and gives us 4 electrons, as well as oxygen gas. This is the point in photosynthesis where the oxygen gas is produced. So if you think back to that big equation that I had written in the previous segment, you notice that one of the products of photosynthesis is oxygen, and it's at this point where that oxygen is produced. So some of these electrons, will head back in here to replace the electrons that this chlorophyll A had to give up. So now we have electrons going along this electron transport chain into photosystem 1. Now what has happened is, light has also hit photosystem 1, and the same thing has happened. We had our accessory pigments, our chlorophyll B and our carotenoids, absorb that energy, forcing the electrons in chlorophyll A to get excited and shoot electrons out of here as well. And those are going to go, the electrons that are coming out of photosystem 1 are actually going to go into a different electron transport chain. And what's going to happen here is, we have something called NADP plus, and we have a proton out here that is going to accept these electrons, and this is going to be converted to NADPH. okay? Now, this is the NADP here is typically referred to as the final electron acceptor in the electron transport chain. That is sometimes a question that may come up, so just know that one. And so now we have this NADPH that's produced here in this step, and that's going to go into the Calvin cycle, which is what we're going to be talking about next, our Dock reactions, or the Calvin cycle. Okay, so now what is also going on is we have a situation where we have, there are some protons hanging out in our stroma over here, okay? And what has happened is as these electrons were going from photosystem 2 to photosystem 1, they were releasing energy, okay? And that energy was able to take these protons and channel them in so that now they're inside. So we have all of these protons now inside here on the interior membrane in our lumen. And this has created a concentration gradient. So where we have this concentration on the inside, the concentration of protons on the inside is greater than that on the outside. Now, we have this enzyme over here called ATP synthase. And what this does is a couple of things. It acts as a carrier protein it absorbs these, it takes these in, okay, these protons, and allows them to channel through here, okay? And it also, I'm just going to rewrite this, just think we're running out of space here. It's getting a little convoluted. I told you we were going down the rabbit hole later, so here we go. I'm just going to write ATP synthase over here. Okay, so this has taken in all of these protons here, channeled them through. And what's going to happen here is this ATP synthase is actually an enzyme that's going to be able to convert ADP and a phosphate to ATP. So just to jog your memory just a little bit, ADP, adenosine diphosphate, and when we add a phosphate here, gives us ATP, adenosine triphosphate. Okay, just want to jog your memories a little bit there. And this ATP is actually also going to go in to our Calvin cycle, our Dark reactions. So one more terminology that I think is important for you to know is this concept that we were just talking about here, where you have a concentration gradient coupled with the ATP formation is actually called chemiosmosis. Okay, so that's, refers to this, like I said, combining with ATP formation here. So that's also a term that might pop up for you. I just wanted to draw your attention to that. So just going to briefly, again, run through one more time, because I know this is, a little out of control, I guess, at this point. When you just look at it, you're like, oh my God. Okay, so we have our light-hitting photosystem 2, the chlorophyll B and carotenoids absorb that energy, pass it on to our chlorophyll A molecules, and the electrons in chlorophyll A get excited and shoot out of photosystem 2, and those electrons are passed along the electron transport chain along to photosystem 1. And what has happened in the meantime is we have water breaking down to protons, electrons, and oxygen gas, and because these electrons in the photosystem 2 need to be replaced, okay? And the electrons that had gone through this electron transport chain are able to replace electrons that were lost in photosystem 1 as light hit that, and the electrons had to shoot out in the same way. And these electrons go into, as I said before, a different electron transport chain, which the end result will be this NADPH that will be used in our Calvin cycle. And in the meantime, this energy that has been created as these electrons do go through the electron transport chain allows these protons to be brought into the interior of the membrane here. And we have our guy ATP synthase that's going to channel these protons through to the outside. And this enzyme is also going to bring these two guys together to give us our ATP, which will also be used in the Calvin cycle. Now, one last thing. A lot of times, sometimes, I don't know, people get confused when they see, well, this is photosystem, why is this photosystem 2 and this is photosystem 1? Because you started over here, and is that a mistake? What it is this was discovered first, so that's why it's named photosystem 1. Because I guess they felt this wasn't confusing enough that they, personally just changed. I would have just changed the name to make this all less confusing for everybody. But, so that's to clarify that. So don't let that confuse you. It's not a mistake. This is photosystem 2 and that is photosystem 1. Okay, so I think we managed to cover everything in the light reactions. So stay tuned for the dark reactions, or the Calvin cycle. Okay, so let's talk about those dark reactions. I know you're all excited and ready for this. Those light reactions, you found those totally fascinating, so more fascination to come. So dark reactions, also known as the Calvin cycle, and this is taking place in the stroma, which is that fluid substance outside the thylakoid that we talked about earlier. And again, we're inside the chloroplast. Okay, so an important concept for you to understand, just big picture stuff, what's going on, is this concept called carbon fixation. Carbon fixation. And that is the conversion of carbon dioxide into an organic compound. So this is sort of big picture that I just want you to keep in mind. Okay, so let's get started. So first, what we're going to look at is we have this carbon dioxide coming in from the air, and this is going to combine with something called a ribulose 1,5 bisphosphate. Now, you might not have to know that name. You have to kind of go with what your teacher expects. Usually they're okay with you just knowing it as RUBP. But what I do want you to know is that this is actually a 5 carbon. Here. Go. It's 5 carbons, and actually we'll do so it makes it, this is why, just sort of side note, I said it was a ribulose 1,5 bisphosphate. So they call it that because we have a phosphate connected at the first carbon and the 5th carbon. But that's a side note that you probably don't have to know. So, okay, so we have this combining with, we have what actually puts these two together, the carbon dioxide and the ribulose 1,5 bisphosphate, is an enzyme called rubisco. Again, you may or may not have to know the name of it, but do know in general, I think that's an enzyme that is bringing these two guys together. Now, what will happen initially? You see I have 5 carbons here, and we have 1 carbon from our carbon dioxide. So that's going to form something with 6 carbons, but it's very unstable. So what's going to happen is it's going to end up forming 2 of these phosphoglycerates, which have 3 carbons each. So we have 2 of these guys here. And what's going to happen now is we have some ATP coming in, and what happens is that these ATPs are going to donate a phosphate. Each going to donate a phosphate, leaving us with 2 ADPs, because again, as you recall, ATP is adenosine triphosphate, ADP, adenosine diphosphate, okay? But as soon as this happens, we have NADPH coming in, and we say it's reducing it. And just sort of some chemistry there, I'm not sure if you remember this, when we reduce something, it means it gains electrons, okay? So that's essentially going to take those two phosphates away and leave us with NADP plus, and then the two phosphates. This PI, if you see that, it's just kind of a fancy way of writing inorganic phosphate, okay? So now we have two of these guys, two P gals, which is glyceraldehyde 3-phosphate. So what happens is these also have like 3 carbons each. I'll write it in the same. So it's going to look like the same, but just know that it is actually different. Structurally, it's different, but at this level, you don't have to know exact structures. I just am more concerned that you understand that we have 2 3-carbon things happening here, okay? But no, that these structurally did in fact change, so just don't get confused there. And now, at this stage of the game here, these P gals can be used to make organic compounds. I'll just put, example, glucose. But, like we talked about earlier, it doesn't have to be glucose, but we do usually use that as the example. And this cycle needs to, continue on. So some of these P gals are going to be used to make more of the RUBP. And this, of course, will need some energy to do that. So we use some ATP here. Now, I just want to play around with the numbers a little bit for you to make this a little bit clearer. If I were to say, if we were to start with the 6, so basically so if this is 6, we're gonna double these guys. These all get doubled. Okay. So, well, not doubled. I'm sorry. I multiplied by 6. So you get just 12 of these. 12. 12, 12, and 12, and 12 again. And this will be 6 and 6. All right. So, reason I did this is if we follow this along here, okay, we're going to end up with, when we use these numbers, which if you go back to that big equation that we did at the very beginning of this video, and you look in that equation, you notice that we do use the 6CO2 as a reactant, because in our product, we are using glucose. And in order to make this glucose, okay, it's actually what's going to happen here is 2 of these P gals, okay, are actually going to be used to make, so 2 of these are going to be used to make the glucose, okay? Because glucose has how many carbons? 6. So 2 of these can now go to make the glucose. And the other 10 are actually going to be used to make the ribulose 1,5-bisphosphate. So I just wanted to show you just numbers-wise how it could work out where we're actually getting, our molecule of glucose. So hopefully that has made things a little bit clearer for you. Because again, if you count up all the carbons here, right? I said we were actually going to, let me actually write this, so 10 of these are going to go to create more ribulose 1,5-bisphosphate. So, because if we had started out with 6 of these guys, right, 10, we want each of these have 5 carbons, right? So this works out when we take 10 of these P gals, right? we end up with 30 carbons, and 6 times, if we say 6 times 5, we get our 30 carbons. So it sort of all evens out there. It all checks out. So that's why I wanted to go through and use these actual numbers just to make it a little clearer for you. So big picture here, like I said, carbon fixation happening, conversion of CO2 into an organic compound. Our CO2 comes in, binds with the help of an enzyme, Rubisco binds with our UBP. We get our PGA, the ATP comes in, adds phosphate, which the NADPH will then take away, and we call that, this is being reduced right here because it's gaining electrons, which will give us our PGAL. And our PGAL can go on to either regenerate this cycle, keep it going, and create more RUBP, or it can be used to create an organic compound like our friend glucose. Okay, so just one more thing that I think it's important that you are aware of that we haven't talked about yet. Although some of you, you might have had this question, sort of popping up. So, we say that we have CO2 coming in, and we know we have O2 going out, right? But how is it getting out, right? So just to clarify that a little bit, basically we have our, I apologize, I'm such a horrible artist. So we have our leaf, or football, I guess whatever I call it today. But today it's a leaf. So on the underside of this leaf in here, okay, and of course why I didn't use the green to draw the leaf, okay. So we have these things called stomata. It sounds like a scary movie, but it's not. It's just these are pores on the leaf that allow for gas exchange. And what I mean by gas exchange is, CO2 comes in, O2 goes out. Now the CO2 we just talked about, remember, so I just want to jog your memory a little bit of this O2 we actually produced in our late reactions. So I just wanted to draw your attention to that, and you might see this, the singular is actually stoma. So this is like the singular. It's one of these pores. It's just called this is the singular. And so this would be the plural. Now, a lot of this is a tough one, right? And I'm glad this is up here to show you that, because this will come up. It's like, is it the stroma or the stoma? So stroma is that fluid surrounding the thylakoid inside the chloroplast. Stoma is a pore, or stomata pores, on the underside of the leaves that allow for the gas exchange. Okay, that's it. I hope this was helpful.